Okay, it's my pleasure to chair the session on the topic of general. This is the general session. Everybody else. And the first talk is by Sudhir, Kumar, Phylomedicine. Sudhir from Temple.
Okay, so I guess I'll give a very general talk of some type. So I'll talk about phylomedicine. This is the research we've been doing for a while, and it has to do with phylogeny and how long-term history of species, not just what happened in human evolution during the last few 100,000 years, but even before. How does that inform medicine, and what can we do with this information that many of us are discovering all the time? And so, as you might imagine, we very heavily use the tree of life. This is one of the trees that we recently built, was published only last week with 50,000 species. So I'll get started and sort of give you an idea that what I'm interested in is an interface of genomics, evolution, and medicine. So for us, genomics is the most important subject matter because a lot of diseases, a major component of those diseases, genomes, based one way or the other. Now, of course, not every part of every disease has to do with genome in full way, but in different partial ways, Mendelian, simple diseases, or complex diseases or cancers, genome has a lot of role to play in our predisposition to that. So that's what the area that I'm in. When you think about genomics, the best thing is technology is incredible. And that means that we constantly discover that we are all different from each other, even more so than we thought before. So each one of us carry millions of variants or single substitutions or mutations in our genomes, and this number is increasing at an exponential rate. So if you look at it today, you have over 200 million SNPs in the non-coding regions and tens of millions of SNPs in the coding region. Exome sequencing is done in cancer and so many other cases routinely now. So we know we have so many variants. And so when I think about these variants, I always think as like a scientist, long time ago I had a gene, had a few mutations, was very happy. But now what we have is a universe of variation. And what we have to do is somehow make sense of it. Some of these variations would have impact on our health, drug response, but many of the variants probably are strictly neutral in a person's lifetime. So how do you distinguish that between them? And so even though I've been doing this research for many years, a few years ago I thought, it's more like what people in the case of physics and astronomy had to do. They had all these stars, these huge constellations, and there was no way to go to them, no way to go out and touch them or land on them. But we had to somehow get an idea of their mass, their intensity, their composition. We didn't really actually have to go there. So similarly, can we do something like that? And I think that sort of a thing is a telescope, which I think long-term evolution can be one for all of this amazing constellation of disease mutations that exist potentially in our genome. So what you have in a telescope are really two parts to it. You have two lenses primarily. And I think evolutionary conservation, which a lot of you know really well about in this community, is something not new, can be one of the lenses that gives us an idea of what evolution has permitted in the past, whether it is human history or whether it is species history. And divergence, what has it allowed to go on and change. And so these two types of information, I think, give us a lot of tools to model methodologies, ideas to tackle understanding of these tremendous constellation of variation that we see around us. And so a few years ago, I named that phylomedicine. Believe me, I thought somebody would have already called it that, but after much Googling, I found nobody used the word phylomedicine. So one good thing about Google is you can title your stuff differently from other people. So we call it phylomedicine. This also separates you from us from evolutionary medicine, which is a name that's becoming sexy every day after so many years of hiatus. And And anybody who's an evolutionary biologist doing analysis of any gene with any disease propensity wants to call themselves, and maybe rightly so, I'm an evolutionary medicine researcher. Well, I don't know what that means because they're not translating. So for us, I thought phylomedicine has to do very strictly with the variation that humans have and how do we diagnose and understand this variation. So it has a translational angle. So in my group, this is an area that I think a number of our laboratories, all of you guys also doing, is we follow a P4 paradigm. And the idea here is to take a very strict approach to research, very systematic approach to research in this area. And so our model is to look for whether there are evolutionary patterns or long-term signatures that differentiate between disease and non-disease variation. And the different types of variation exist for disease, and you want to be able to test what are the limits of evolutionary analysis to understanding and diagnosing these variants. What are the actual theoretical underpinnings? We have to have a null hypothesis. We can't have a knee-jerk reaction that everything has a reason to be there, or it was so before, it's so now, it must be something. So those stories we have to go away from and truly build a null hypothesis. And in genomics, that's possible to do. Many fields, it's very hard to do that. In the case of, if you have a null hypothesis, you have a system, you can actually make predictions. And then the question is, can we use molecular evolution over long term as a way, as a tool to diagnose mutations? And once you make methods like these, we have to develop products so people can actually use our methods rather than just have them published and be happy. So that's another area. So this is the P4 paradigm that we follow. I'll very quickly go through this paradigm for you. I can speak very, very fast, but I'll keep it slow. So let me give you some very simple results, since the community here is very diverse, from physicians who haven't done molecular evolution and evolutionary biology from DNA and genomes all the way to experts in evolutionary biology and molecular evolution. So the first thing is, are there patterns in disease variation that distinguish them from non-disease variation? That's a very straightforward question. And so what you see here is a bunch of black dots and a light blue line. So the black dots are about basically the data. When you look from left to right, on your left to right, is left side is extremely conserved positions, and right side is positions that change a lot in evolutionary history. So what you see immediately is most of the mutations that are disease associated, Mendelian in this case, which is a very strong high penetrance mutations, simplest one to study actually, they are in fact overly abundant at slow evolving sites. So evolutionary conservation, if a position is really important, is practically never changed in the history of human to fish. This is where you're going to find disease mutations. This is actually interesting. And so and this trend is really goes very quickly. So it's not like there's a huge number of disease mutations found at very fast evolving sites. So evolutionary information coming from species gives you a trend, which is distinct from the trend that you observe when you look at the mutations that are found to be not have any disease connection, or at least any Mendelian disease connection. So that's an interesting result. It separates the two. Are there any other such signatures? Well, before we do that, we have to test our hypothesis properly. So we do that by observing our expected ratios, and you can see the overabundance. I won't dwell on it, but we found this pattern, I don't know, 13, 14 years ago, first time. And a lot of people have worked in this area over the last 15 years. And so this is a very interesting pattern that constantly comes back over and over again. And this is a pattern when you actually do the same analysis with non-disease variation. So seems like very distinct, completely opposite patterns. And so it makes me feel good that evolution could help with diagnosis of these mutations. Now you think about sort of what kind of mutations or variation are these at amino acid level that are involved in disease. Are they really distinct from non-disease variants when it comes to their biochemical properties that are not evolutionarily derived in any way? They are to do with, for example, here, Grantham dissimilarity biochemically that has to do with size, polarity, charge of amino acids. And what you find is that disease variation is 50% more severe than non-disease variation, which is a nice result. So if this is so, what about species? It's very interesting. If you look at variation found among species of the same positions even, what you find is it's almost the same as human non-disease variation. So if I did not have any human polymorphism data at all, I just look between human, chimp, gorilla, orangutan, given all of the species, I'll actually be able to predict what would be a benign variation in our genome potentially. So that also looks very positive outcome. And interestingly, in 1980s, starting in 1960s actually, in the field of molecular evolution, a neutral theory of molecular evolution was put forth by, or really championed by Moto Kimura. She showed me a 3. Okay. And so essentially that sort of explains, said this was what we observe. That's what we see. And so I would like to make a case here is that if we know information about species, we can actually tell something about human variation, individual variation, not just populations. And then also, of course, species information is good for population information. And interesting thing is about human history is so short, couple of 100,000 years maximum, that we can actually estimate all of these parameters quite independent of human variation. So we can actually have no circularity argument issue, because remember, human chimp is 5 to 7 billion years, and human history is only 0.2. So it's a big difference in the scale. And just for those of you who think the complex diseases don't have anything to do with long-term fecundity and stuff, yes, there's a lot of that literature out there. We published a paper a couple of years ago showing that, in fact, if you look at the positions in their retention over time, we actually have a very distinct pattern between disease variants that are from GWAS, that are supposedly causal, and non-disease variants. So similarly about odds ratio, the effect size of the variation. So we do make products, as I said, we should. So we developed some new diagnosis methods that I call evolutionary diagnosis or EvoD methods in this area, which take only evolutionary information, no other black box approach that many times many support vector machine and all those kinds of things do. These are very straightforward evolutionary information used to generate pretty good accuracy. And finally, we have to make some products. So you see, we have software called Mega MD. MD, of course, uses mutation diagnosis, not a doctor. It can help you with more than just mutations. And that's available online. And so I don't have time to talk about a lot of other things here, but I want to conclude by saying that from our analysis, from what we are seeing in complex diseases and others, that an overwhelming majority of disease variants will disrupt fundamental biological function. And they will actually disrupt neutrality of alleles and positions, and which is really equivalent to purifying selection. So that's primarily what the main narrative would be. It doesn't exclude adaptation, but that will be, as expected in the past, a smaller narrative. So that is another thing that I'm seeing, and I didn't have time to talk about it today here, is human-specific adaptations over the last 100,000 years or 10,000 years or 5,000 years would actually have some impact, but they would actually be minor role in genomic medicine. And if you want to know more about exactly what I mean by that, we published a perspective article a couple of years ago where we talk about the concept of previously neutral and previously adaptive changes and how that impacts and explains available variation, including all cases of adaptation we know today. So I think the future is that because neutral theory of molecular evolution works well, we can truly make very good tools and methods. If it did not work well, then adaptive was the only thing. It would be much worse. And I'm stopping now. For those of you who are interested in joining our enterprise in phylomedicine and others, we have a new institute at Temple University. Thank you very much.